before I really go into, the genomic results and all that, I thought I would just, again, disclose that I'm actually Canadian, and this is kind of what we have to deal with, and we also have to deal with these stereotypes that are very problematic. So, like as you can see here. Now, being from Canada, actually, I grew up in Montreal, and I have a connection to asbestos because, as some of you may know, there's actually a town in the province of Quebec named Asbestos. So it's, there it is on the map, Asbestos. And so just to orient you, Maine is over here, Vermont's over here, and New York is over here. And so town of Asbestos. And this is like a welcome sign right in front of the open pit mine. This was a huge open pit mine. It says, Bienvenue à Asbestos, bon séjour chez nous. So, welcome to Asbestos, enjoy your stay with us. Just to stay on the topic for just a couple of more slides. So this was a site of one of the major asbestos mines in Canada, the Jeffrey mine. And remarkably, this only totally ceased operation in 2011. And even more remarkably, some politicians actually wanted to reactivate the mine in 2012, I guess, to create jobs. But I don't think this was a good idea. So it didn't transpire. So this town, though, has not changed its name, to my knowledge. So it actually, of course, leads to stories like this. Can you attract tourists with a name like this? And this is, of course, a beautiful picture of the now inactive open pit asbestos mine. And if you go on TripAdvisor, it's on TripAdvisor. And TripAdvisor, I guess, automatically populates the web pages. So it leads to things like this: your top asbestos restaurants and top I can't read this. Top asbestos things to do. So it's kind of an odd situation. In any case, getting back to the matter at hand. So I want to tell you about the TCGA Pleural Mesothelioma Project, which I've had the pleasure of co-leading with Bruce Robinson and Peter Campbell for the last few years. So the Cancer Genome Atlas, for those of you who are less familiar with it, was a large consortium type network created by the NCI and the National Human Genome Research Institute in the mid-2000s. And the goal was to provide the kind of a gold standard characterization of all major human cancers. So mesothelioma was actually one of the last cancers to be added to the list. Once they had done the biggies, there was like a discussion of which less common cancers should be done. And at that time, I was already involved in TCGA. This was at that time was 2011 when these discussions about what other cancers should be studied were going on. At the same time, Bruce Robinson was traveling around the world, as I think he does a lot, trying to get a global malignant pleural mesothelioma genomic effort going. So this kind of converged, and we managed to convince TCGA to include MPM as a project, and the project was initiated in 2012. There's some other points in the timeline that are shown here. Finally, we had the first public presentation of the results at IMIG was it April or May, and at ASCO, and the manuscript is in press at Cancer Discovery. Hopefully will be available online shortly. So the tissues came from all over the world. Initially, we managed to obtain 87 cases, and these really had to be pristine, untreated frozen tissues with matched normal. So the specimen requirements were extremely stringent. So we had to exclude 8 cases for the reasons listed. That gave us 79 cases, which then went into expert pathology review. And the results of that review are shown here. So 4 cases ultimately were removed as being more consistent with another diagnosis. Again, showing you that how the difficult the diagnosis of mesothelioma can be. And then 1 additional case was excluded due to very low tumor content. So ultimately, we were left with 74 cases. This was the histologic distribution. And as expected, just to kind of get a sense of whether the data set was representative, the overall survival is shown there, as well as expected, epithelioid cases did better than cases with a sarcomatous component. So this was this is kind of an overview of some of the genomic data, specifically the number of mutations per case, and the samples are arranged here by histology, so epithelioid and sarcomatous component. And one thing immediately to know is that mesothelioma is actually not a highly mutated tumor. It has a low tumor mutation burden, so this is something to keep in mind in terms of immunotherapy. So we found a mutation rate of less than 2 mutations per megabase, except for 1 sample that had an MSI-like signature and an elevated mutation rate. So one of the analyses that one typically does in these genomics projects is to use statistical methods to identify genes that are mutated more frequently than expected by chance. And so we did that. And we also overlapped this with a similar analysis that had been performed previously by Rafael Bueno and colleagues on 99 exomes. And as you can see, there's significant overlap. So these are obviously very familiar genes, except for the last one, which I'll get to a little bit later. So overall, though, again, it's a tumor with a low mutation rate, and the overall number of significantly mutated genes identified in these studies was correspondingly low. So back in 2011, we reported somatic BAP1 mutations, which were, and that report was followed quickly by the first report of germline BAP1 mutations by Joe Testa and colleagues. So this data set allowed us to do what's probably the most comprehensive analysis of BAP1 alterations, because we had mutation data, copy number data, expression data, methylation data, and so on. So the overall prevalence of BAP1 alterations in this data set was 57%. I think we've seen in the literature that the numbers are in this range, like in the maybe 55 to 70% range. And as expected, samples with a BAP1 alteration had lower BAP1 expression. We used many bioinformatic tools to look at what is different in mesothelioma samples with BAP1 loss. One of the more interesting things that was found was that they show a higher activity of a transcription factor, interferon response factor 8, which is suggests that we should be looking at BAP1 status in immunotherapy trials to see if this has an effect on immunotherapy response. In terms of P16 deletions, we know very well that P16 deletions are quite common. But looking at the copy number changes globally in mesothelioma, one thing that's very striking is that there are no recurrent gene amplifications. It's all deletions. So again, a very distinctive pattern. Of course, the losses of BAP1, but also of P16. One interesting thing about P16 that was already known for many years, and here's a paper from 2003, is that P16 is right next to a gene called methylthioadenosine phosphorylase. So it's actually about 200 kb from P16. And more recently, this has become interesting because and this is confirmed that here in the TCGA data, the frequent co-deletion of MTAP and P16, and as expected, when P16 is deleted, its expression is low, likewise for MTAP. And recently it's been shown that MTAP deficiency leads to reduced activity of an arginine methyltransferase called PRMT5, and therefore the cells become more sensitive to inhibition of PRMT5, and there is a PRMT5 inhibitor, AG270, in a phase 1 trial in tumors with MTAP deletions. And actually, if you think about solid tumors, I think mesothelioma and pancreatic cancer have probably the highest prevalence of P16 deletions. So clearly, they would be prime candidates for this approach, for this trial. In terms of significant negative findings, just to remind you that we also confirmed that there are no, none of these canonical, let me rephrase that your typical activating mutations in MAP kinase signaling are very rare in mesothelioma, so we did not find hotspot mutations in receptor tyrosine kinases or RAS-RAF genes, although these have been rarely reported in mesothelioma. We did not find any fusions involving EWSR1 or RTKs. There have been reports of that. And we did not find any SV40 sequences. This is something that my lab also debunked many years ago. One unexpected finding was that there were 3 cases. So this is the copy number plot. I'm sorry, the LOH analysis of the samples. And there were 3 samples that showed loss of heterozygosity of almost the entire genome. So greater than 75% of the genome showed loss of heterozygosity. That's shown here, these 3 cases. And the other interesting coincidence was that these were the only 3 cases in the data set that showed mutation of SETDB1. And all 3 also showed mutation of TP53, which is relatively uncommon in mesothelioma. We had access to a Japanese data set through collaborators where they had also observed this phenomenon. They had 2 out of 74 cases, and again, both with P53 and SETDB1 mutations. These are the allelic copy number plots for these cases. And they had a very, some kind of characteristic pattern whereby chromosomes 5 and 7 were often the only regions of the genome that did not undergo loss of heterozygosity. This is something that was observed first by a Korean group in 2016. They reported one patient with TP53 mutation, ZTB1 mutation, and widespread allelic loss. And if you read the supplementary materials of the 2016 Bueno study, they note that in a few samples we observed widespread LOH. We worked with Jonathan Fletcher, cytogeneticist at Brigham and Women's Hospital, who had accumulated a large cytogenetic series of mesotheliomas, and he had noted this phenomenon in 16 patients. So these karyotypes, as you can see, are haploid, and they also had, unlike mesothelioma in general, they had a kind of a 50-50 male-female proportion. He was able to test 3 cases by Western blot for ZTB1, and all 3 were negative. And he also noted that they had a high prevalence of TP53 mutation. And here's an example of such a karyotype. As you can see, loss of 1 copy of essentially all the chromosomes except 5 and 7. But what we see in a lot of cases is that this then undergoes reduplication, so that you once again have 2 copies of each chromosome, but they're actually the same 2 copies of the chromosome, the copies of the same chromosome. So in summary then, this seems to be a novel subtype, a novel genetic subtype of mesothelioma. Accounts for about 3% of cases. Again, over 75% LOH, chromosomes 5 and 7 typically spared, has a characteristic mutation profile, seems to be slightly younger. There's actually, if you look at the Jonathan Fletcher data, there's even a female preponderance. We did not see a difference in distribution of histological subtypes. I can't really comment on asbestos association. The hypothesis is that ZTB1, which is a histone-modifying protein, may cause derepression of certain monoallelically expressed genes, thereby making these viable. And most recently, it's very interesting to read that Hürthle cell carcinomas of the thyroid show the same LOH pattern in actually the majority of cases, and that includes sparing of chromosomes 5 and 7. Moving on, so since we had multiple types of genomic data on these samples, it's possible to do what's called integrative clustering, so using data from every platform to identify subtypes of mesothelioma. And this was done using an algorithm called iCluster that was developed by Ronglai Shen at our place. And this generated 4 distinct clusters. And this clustering then was based on a combination of DNA copy number, methylation, mRNA expression, microRNA expression, and long non-coding RNA expression. So if we look at this more closely, these are color-coded clusters, and they actually range from the most favorable to the most unfavorable. And these are some of the characteristics of these cases. As you can see, the most unfavorable had a very high Epithelial-Mesenchymal transition phenotype, so more of the sarcomatoid cases. Low mesothelin, and this was due to methylation of the mesothelin promoter. High aurora kinase, And this is something that was actually also previously known. So we had actually picked up the same thing back in 2006, that the most aggressive cases of mesothelioma show overexpression of aurora kinase. Whether that's really targetable is another question. P16's been known as a negative prognostic factor for many years as well, shown by multiple groups. So when we did the survival analysis, again, you can see these are ranked from the most favorable to the most unfavorable, and showed very nice separation of these survival curves. And again, this was not, the clustering itself did not include, was not based on survival data. In a Cox regression, the integrative clustering was independently significant in a model that included histology, of course, and P16 status. And this was repeated using only the epithelioid mesothelioma cases, because the clinical argument was, well, we already know that sarcomatous mesothelioma is a bad actor, and we obtained similar clusters. And the signatures were reduced to mRNA signatures and were independently validated on separate independent cohorts. So further examination of these clusters and what defines them might identify either predictors of prognosis or targets. Now, one of the analyses, and a gazillion analyses were done. One of the more interesting analyses was an analysis of EMT signature. So this was based on mRNA expression profiles, and was compared to all the other tumors in the TCGA data set. And interestingly, maybe not unexpectedly, mesothelioma has one of the strongest epithelial-mesenchymal signatures in this set of cancers, second only to, of course, sarcomas. Now, because EMT has been associated with differences in expression of immune response molecules, an analysis was done looking at how EMT score in mesothelioma correlated with immune checkpoint gene expression. And one of the kind of unexpected findings was that this immune checkpoint molecule called VISTA was extremely significant in terms of association with EMT, in the sense that it was highly anti-correlated with EMT in mesothelioma, implying that it was higher in epithelioid cases than sarcomatoid cases. And when we looked at VISTA expression across the entire TCGA data set, mesothelioma actually had an outlier expression of VISTA significantly higher than any other cancer that had been studied by TCGA. So VISTA, and this is out of my comfort zone. I'm not an immunology person, but VISTA is a relatively understudied immune checkpoint molecule, structurally similar to PD-L1. One of the aliases is PD-1H, and this is what it stands for. So it's known to suppress T cell activation, much like PD-1 or CTLA-4. And its expression in mesothelioma, of course, is quite interesting. We looked at this, so at the mRNA level, VISTA is most prominently expressed in epithelioid cases, much less or not at all in sarcomatoid cases. And these are examples of VISTA immunohistochemical staining in two cases. Case one being one of these dots up here. This is a high power showing very pronounced VISTA staining in these cases. And it was also highly correlated with mesothelin expression. So this all goes along with VISTA being expressed in better differentiated mesotheliomas. It also is expressed in a benign mesothelium, so shown here. And this is reactive mesothelium that's folded over here. It's also positive. And this was, we confirmed this in multiple samples of non-neoplastic mesothelium. If you look at mesotheliomas in terms of VISTA expression versus PD-L1 expression, there's an interesting almost reciprocal relationship. So VISTA's highly expressed in epithelioid meso, as I mentioned, and that expression is typically strong, whereas PD-L1 tends to be, when it's positive, tends to be weakly positive in epithelioid mesothelioma. If you look at how this plays out across the different subtypes of mesothelioma, you can see the decreasing expression of VISTA and the increasing expression of PD-L1 as you go from epithelioid to sarcomatoid. So in all, in the immunohistochemical study, which I should say was not part of the TCGA analysis, is a separate analysis, 85% of mesotheliomas are positive for VISTA, whereas only 38% are positive for PD-L1. So we postulate that since non-neoplastic mesothelium expresses VISTA, the expression that we see in epithelioid mesothelioma may either reflect retained expression of a normally expressed molecule or may be possibly selected for by immune pressure. There have been reports of VISTA expression as a resistance mechanism to other immune checkpoint inhibitors, and it's certainly tempting to speculate that the generally better response of sarcomatoid mesos to immunotherapy may be due to VISTA expression by epithelioid mesos. And this provides, of course, a biomarker and a rationale to look at anti-VISTA targeting in clinical trials, and there are agents that target VISTA that are in various stages of clinical development. Actually, just on Tuesday of this week, some of these results about VISTA were presented by Marjorie Zotter at the World Lung in Toronto. So with that, I'll be pleased to take questions. This, of course, being a TCGA project, is an enormous joint effort by many, many people, the key ones being listed here. Thank you very much.